Hi everyone, Dr. Brazier here, and I'm really excited to present one of the last videos over the plant kingdom that we have been talking about the past few weeks. This is going to be lab number 9, Angiosperms, Form and Function. And as you remember, we did start with, and we have looked at this plant cladogram every week over the past few weeks. And we started with this ancestor here, which is green algae, right? This is a monophyletic group, which means that they all come from a common ancestor. And we're going to look at how we went from these very simple, seedless, non-vascular plants, added vascular into it, and then essentially added a naked seed. And now we are at the grand papa of everything else, if you want to say, or the pinnacle of the most complex plants that we're going to look at, which are the angiosperm. And so angiosperm, which is phylum Anthophyta, are the most successful groups of plants today. They arose about 124 million years ago, and they are called angiosperm because they're flowering plants. And their ovule are always enclosed in diploid tissue. They're not naked. Their dominant plant body is the sporophyte. And they're critical innovations for this group, because we've talked about that every single time. They're successful because they have the vasculature and the flowers, and specifically the fruits that are enclosing the seeds, and that they have developed an endosperm. And an endosperm is part of the seed which acts as a food store for the developing plant embryo. It usually contains starch with proteins and other nutrients. Now, we've looked at this, and just to kind of get an idea of all the different groups that we've looked at. And so when you go through here, you should be able to easily talk about the 4 major classifications, know what phylum are associated with them, and the examples that we use in class. You should be able to identify them. You should be able to know things like what's their dominant plant body, how do they reproduce, those kinds of things. And so we've started with something that is very simplistic in nature compared to the rest of them, the seedless non-vascular. Marchantia, the liverwort, was an example of that, hornwort and moss. These are known as bryophytes, right? And as we moved forward, we became more and more complex, going to seedless vascular, gymnosperms, and now we're at these angiosperms. It is phylum Anthophyta, and we're going to be looking at monocots or dicots, or which is used more commonly now, eudicots. And so the 2 main types of angiosperm that we are looking at, like I said, monocots and dicots, or eudicots. You can tell from basically their early development stage all the way through to adulthood which type of angiosperm you have. If you look through here, you'll notice that if you have a monocot, and you can look at the root of the term, because mono is standing for one, and the cotyledon is basically where the cot comes from. So this is just a single one. And so cotyledon is an embryonic leaf in a seed-bearing plant, one or more of which are the first leaves to appear from this germinating seed. And so if you have one of those, it's a monocot. The eudicot or dicot will actually show 2 of these. Now, if you're looking at the formed plant, you'll be able to see in a monocot it has the long, narrow leaves and parallel veins. These are the lines that are going down the leaves. If you look at the eudicots, they have more of this broad leaf and a network of veins. When you look at their vascular bundles, you can see over in the monocot that they are scattered, where in the eudicot or dicot, they're bundled in rings. And then for their flowering, if you have a monocot, they're generally in multiples of 3. And when you're looking at dicot, they're generally in multiples of 4 or 5. Now, when we look at these plants, all flowering plants, aka angiosperms, feature 3 vegetative organs. They feature the roots, the stems, and the leaves. The roots are there to primarily anchor the plant to the soil and to help to absorb nutrients, where the stems support the leaves while conducting nutrients and water throughout the plant body. And finally, the leaves are going to carry out photosynthesis, and that's going to help sustain the plant and the ecosystem. Photosynthesis, which you learned about in the first part of general biology, for plants, they need to essentially utilize one of their fundamental building blocks that they need is carbon dioxide. That is what helps them create sugar through an enzyme called ribisco. And so they are able to incorporate carbon dioxide into a molecule to essentially make sugars. Well, that helps because its byproduct of that process is oxygen, which we intel or animals will use to essentially create energy through cellular respiration. So it's like a symbiotic relationship. We give them what they need, they give us what we need, and things work out really well. Now, these plant organs are made up of 3 types of tissues. And so you see we have ground tissue. Ground tissue is tissue that makes up the interior of the plant organ. It's going to perform various functions like photosynthesis and storage, and it's composed of mainly 3 kinds of cells. You have the parenchyma, the collenchyma, and the sclerenchyma. So the parenchyma, these cells carry out most of the photosynthesis and help store carbohydrates. The collenchyma are cells that usually have thicker cell walls and provide structural support in those plants that still have growing regions. And the sclerenchyma are usually those cells that are dead at maturity and make up that thick woody part that we can see in plants. So what parts of the plants have these? Do the roots just have one kind of tissue? The stems have another? The leaves have a different one? The answer is yes, they basically have all types of tissues throughout the entire plant. It doesn't matter where you're looking, you're going to have dermal tissue, ground tissue, vascular tissue. Dermal tissue, ground tissue, vascular tissue. Dermal tissue, ground tissue, vascular tissue. So they can be incorporated within the different objects of the plant. They can look differently. They can be arranged differently, but the tissue is there throughout. And this just kind of gives you a quick look. I'm not going to go into a lot of detail, but you can see here each one. This is a parenchyma cell, and that's important. You can see all the chloroplasts that are in there. Those are little plastids that allow the plant to take energy from the sun through the molecule known as chlorophyll. It's a green pigment, and essentially it is going to absorb those particular electromagnetic radiation waves to then the plant is going to use to create carbohydrates. And then you can both see the sclerenchyma that's here, which is the woody part, and the one that's still growing, the collenchyma there, that's supportive. Of course, they'll show you pictures of what it looks like for the xylem. And xylem is important because it helps to conduct water, and phloem, which is here as well, is important because it helps to conduct through the organism organic compounds like sugar. So now that we're here, let's talk about the anatomy of the plant. As you can see, we have two different systems when we're looking at a plant. We have the shoot system, which is the part that's above ground. That's going to include the stems and the leaves, and that's where all photosynthesis is going to come from. You have the root system down here, which is going to anchor it and allow for water to be absorbed and those nutrients that you need to get from the ground. And then basically just identifying, looking at different parts of the plant. You have the roots, again, to help anchor and to give you nutrients. It's going to flow up through the stem, which then, if you look on here, the petioles are this area that you're going to get where the leaves are. And now the leaves are here, and when we're looking at the leaves, you can tell different areas, and we're going to go over these again, right? The blade that you can see here is going to incorporate what we have. This is the midrib. On the bottom here is the base, and then you have all the veins that are coming off here. The end of the leaf that's there is called the margin, and the top here is the apex, and all of that is going to essentially be the blade. The petiole is going to be this area that connects it right there. Now, between the leaves, you can have what's called internodes, and where the leaves are, the little growth there are the nodes. And so when you look at this, the internode, the node, and these actual axillary buds that you get, those are called a phytomere, which is a functional unit of a plant. Now, what comes off of the stem here are the branches that come off. So I think I misspoke a second ago. I think I called this the petiole, which is not the case at all. I'm sorry if I kind of made anybody get confused. That is going to be that attachment right there on the side of the stem. And then the buds are going to be based on, you can have an apex or a terminal bud, which is here at the top. And then you have on these nodes where they're axillary buds. And these are also known as lateral buds as well. And different things will grow. And actually, the terminal bud will determine if the other buds function, if they are the apical or terminal bud that's there, releases a hormone that can actually prevent these lateral buds from growing or cause them to grow slower, to let you know. So I hope that helps a little bit. I know you go over this a little bit in in General Bio 1. And this is just kind of showing you so that you can see the differences between a monocot and a eudicot. And so you can see the flowering is different, the way that the leaves look, how you have very prominent petioles on the eudicot. And so I just wanted to show you so that you could look and kind of get an idea of the structural differences when you're looking at these. So let's now go through and we're going to hit on every single part of the plant that we are talking about, particularly every single kind of vegetative organ so you can understand it a little bit. So the roots, which is this nice picture here that shows this is like a taproot. They anchor the plant to its substrate. Again, it helps to absorb water and inorganic substance. And essentially, it conducts the above upwards to the rest of the plant production and the meristem. And this is caused by certain hormones that are transported to other parts of the plant. And so when we're looking at a meristem, the growth of the plant occurs in this region. It's a region of plant tissue that's found chiefly at the growing tips of the roots and shoots, and basically in the cambium, and consists of actively dividing cells that will form new tissue. It is the production of where the production of secondary metabolites are held, and it has importance in the storage of nutrients such as carbohydrates or fats or lipids. And so the origin of the root, you have what's called the primary root, which is the very first root that forms in and emerges from the seed. Then most other plants, the primary root then develops into a taproot, which is that large central root. From the lateral root emerges. And so when we're looking at the root tip here, and this is all in here. If you'll notice, the beginning of the lab doesn't talk about any of this, but I wanted you to understand a little bit so that as you went through and you were identifying different models, that you did understand what we're talking about. So this is the structure of the plant root tip, so the very end of the root tip. And so it can be divided into zones or these segments. And so you have here, which is the root cap, and this is what's called the apical meristem. And this is where you're going to essentially, the primary plant body grows by the activity that's at the apical meristem here. And then you have what's called here the zone of elongation. And so this is the rowing cells of elongation. This is a primary meristem. And up here is the zone of maturation. And this is where cells mature. They're no longer elongating. They're pretty stable. Now this tip or cap here, the root cap, is actually protected by a layer of live parenchyma cells down here. And basically when you're looking through here, you can see the different areas. You have the inside, which is the endodermis, the epidermis on the outside. You have in between, you have a little bit farther, as you get into the center, you have the pericycle. And this is just the secondary lateral meristem that gives rise not only to the side branch roots, but is unique to the root itself. So anytime you see a meristem, you just think growth, right? And then in the side, you can see where it becomes vascular. You have the xylem here, and then you have the phloem here. Can you tell me what the xylem does again? It's important in moving water, and the phloem is important for organic substances. Right? Now, what's cool also is the root cap. Not only is it important in kind of the growth of the root and stabilizing it, but it also contains statocytes, which are involved in gravity perception of the plant. So it grows in a certain way based on the pull of gravity, which I think is pretty neat in itself. Now you can identify the difference between a monocot root and a eudicot or dicot root by how its structures are put together. So you can see on the outside here, you have the epidermis. And then as you go to the center, here you have circles, which are the xylem, if you see. So you have all these dots around. And over here, the dots are kind of in an X shape. The phloem is that area that's around the circles there. And here you can see it's kind of got this triangle look shape over in the dicots. This is the area called the cortex. You can see that layer that goes around where you find the xylem and the phloem, and we looked at this earlier. That's that pericycle. And then you have that layer around the outside, which is that endodermis. And then like I said, the epidermis is out here. So if you're looking at the histology, if you took these and cut these and you looked at it underneath a microscope, this is what it would look like. And you can see very much that the monocots have what looks like all of these little dots that are here. Those are the xylem. And then the area around it is the phloem. And then you can see from the meristem, or that pericycle is around it. Where this, you have the X that's there. And that kind of area between where the X is, that's where the phloem is. So they are drastically, you can take a bit of the root and you can look at it and obviously see there's a huge difference. And this is just kind of to help you get a little bit closer up of what it looks like, so you can see that phloem. On here, they're showing you that the xylem is over here, and you can see it's kind of in this area that's here, and then fibers, and then that phloem that's there. And then, of course, that layer of cells that happens around them, and the epidermis. Now, besides the roots, specifically just looking at the roots, we can look at throughout the plant, because we did talk about the root apical meristem, but that is not the only meristem that's there. You have the lateral meristem that we saw down below in the roots that they were talking about was more of that. If you go back here, remember that pericycle that we were looking at? That goes up and continues on up, and then you have the shoot apical meristem. So the apical meristems are the primary growth that you see. They're the lengthening, making it longer. And so you can see it, it's either at the shoots or the roots. And they will produce new leaves and flowers and make this plant taller. The lateral meristem is a secondary growth, and this helps to widen a plant. It occurs at the cambium, and it produces bark on a tree, just to kind of let you know that's a perfect example of the two. And so just so that you can kind of look, and we want to get a good idea, what does the root apical meristem look like when we're underneath a microscope? Well, here is the whole root cap that's there, and this at the bottom is the root apical meristem that you can see, right? That you can kind of look at. And they're showing you that all that growth that you're going to get is through here. So you see where this is, and you can see that differentiation, that this is what we're looking at for the root apical meristem. Now, if you were to go to the top and you wanted to look at the terminal bud, or the shoot apical meristem, you would be looking at this. So here's the shoot apical meristem. That is where the growth is going to come out of from there. And when you're looking at the side, they're showing you the lateral meristem here, and that cambium that we keep talking about is right here on either side of where you see that. That's the cork, and here's the vascular that's in the side, and that's where you're going to get the thickening of the stem and the root versus the elongation, which is where you get on both the terminal and the close to the root cap with the apical root meristem. And so if we're looking at this, I wanted to give you an idea of being able to see the different types of tissue that we have, the dermal, vascular, and ground here. So you can see of course, the dermal, think epidermis is on the outside, and you can see some of those root hairs, that's going to be part of that. And then when you look at the ground substance, the tissue that makes the interior of the plant, does things like photosynthesis, storage, all of that, you can see that that's the yellow part. And so in here, you're going to see a lot of what we know as the ground meristem. And then, of course, this that you have in the middle, that vascular tissue is where you're going to see the phloem and the xylem in the middle. And so here they want you to see on the end, this is your root cap down here, right, that you see. And so this area, that's right where the root cap, that you can see that differentiation between the look where it gets darker here and it's lighter here. That darker part is actually the apical meristem. And this center here is where you're getting a lot of that growth coming from. And so this is the zone of division that's here. These is where the cells are becoming elongated, and this is where they're maturing and they're no longer getting longer or growing. Now, the types of roots are dependent on the type of plant. You have the fibrous roots, which is what we're used to seeing when we pick up like some kind of plant generally that you're looking at. You have the adventitious root, which is, if you look at this is the roots that are above ground like that you can see, and then the tap root that you're looking at, like radishes or carrots or anything of that nature. So this looks like a bunch of fibers. This looks like, on the tree, the really thick things that you see, and this is normally really large and in a lot of cases singular. Now, what's really neat here is that sometimes bacteria and roots can be advantageous together. So rhizobium is a gram-negative bacteria, and it does what's known as nitrogen fixation. It helps to get that important building block to the plant. And this happens in the plant roots. It's going to help basically make it grow, and it allows the bacteria to help with sugar and nutrient production. And you can see this happening because when they are there, they will actually cause these like root nodules or like tumors to form. So anytime you see something like this on here, that's actually the bacteria colony working with the plant to create the symbiotic relationship. It's pretty cool, to be honest with you. And so this, I just wanted to show you kind of a close-up of what was happening here, right? So here is the sclerenchyma cells. And here you'll have where you'll actually get a chemical signal here that attracts bacteria, and then this infection thread will form. And so you can see those infection threads right here, right? And so they will go, actually, so let's show what's actually happening in that root nodule. I wanted you to be able to see it a little bit better. So basically you have this root, and you'll see right here you have a root hair, and the bacteria is right there. And so this is what's called an infection thread. The bacteria will actually start becoming, or will infect that root hair. Now from that, a bacteroid will form. And so you can see this is where the bacteria are in here, and they're starting this process of this kind of nodule to be formed here. And you'll see it start to happen in that area for elongation, that pericycle area where the cells start rapidly dividing. And so once you have enough cells there, you'll notice that the growth continues and the root nodule actually starts to form because it starts to kind of come out of this root. Now, over time, this is going to become a well-structured part of the root. So you're going to start seeing vascular tissue forming for this bacterioroid that's here. And then over time, you have the mature nodule. It can grow to times the diameter, many times the diameter of the root, as you saw there. And essentially, you have the bacterioroids that are there. And it's symbiotic. The bacteria are living there, doing nitrogen fixation, allowing to provide those fundamental building blocks for the bacteria and for the plant, and the plant is doing the same. And so this just kind of shows you how when they're doing the nitrogen fixation, right, bacteria cells in the root nodules, basically they can produce things like there'll be nitrogen gas there, and through this enzyme nitrogenase, it will break down that into ammonia, and then you can use from the ammonia, it can break down further into things like amino acids, or even smaller, and be able to use and get vitamins from it, certain hormones, nucleotides, and that will be some of the building blocks that the plants will use. And essentially the plants, using CO2 through photosynthesis, will make sugar, which they'll give back to the bacteria. So this is very much a symbiotic relationship. Now we're out of the roots, we're going to go into the stem system. So a stem is an above ground structure that provides support and facilitates the conduction of water and nutrients throughout the plant. It can be herbaceous, which means it has no wood like in the monocots, or it can be woody like in the stems of the eudicots or dicots. Herbaceous stems undergo a primary growth or growth in length resulting from the activity of the apical meristem located in the terminal bud. There is no increase in the girth of the plant, like in the stems of maize or grass, right? And so when we look at this, this is both giving you an idea of the monocot stem versus the dicot stem. And essentially what you're going to have is you're going to look at a model that kind of gives you an idea of how this works. And so the monocot, you can see that the vascular tissue is all over. They're everywhere, right? And as you go through here, each one of these is going to end up having a xylem and a phloem within here. Now, the sclerecama over here in the eudicots or dicots are towards the outside. And if you remember that is something for woody plants, right? So you're not seeing that as in the monocot, this particular type of structure. And then here you can see on this side, it has towards the inside the xylem, towards the middle right there is the phloem. And the ground tissue is the same for both of them. It's just, it's a nice clear area in the middle where you kind of have towards the outside all these vascular bundles. So as you can see here, this is considered a vascular bundle, and there's a ton of them in the monocot everywhere throughout the stem. And these are towards the outside or towards the epidermis area of the stem. So if we were to take one of those individual, back here, if we were to take one of those individual ground tissues, if you look at these circles here, and then you go back and you look over here, you'll see the same thing in the monocot stem. So essentially what you have is you have the xylem, which is for water again. And so you're looking at these things here. And then the phloem is going to be over in this region that's over here. And you have the parenchyma cells that are around those. Remember, those are for photosynthesis. They have chloroplasts, specifically the molecule chlorophyll, to help get that energy in. And so this kind of gives you an idea of how they're put together. And just to see an actual histology of it, you can see the bundle sheaths there. You can see the vascular bundles. And they're showing you that, where you have that darker area here is the phloem. And essentially the xylem are these lighter areas that you're seeing in here. And so the whole thing here is called a vascular bundle. And the thing that goes around it is the sheath. And this area you can see the pith and the epidermis is on the outside. Now, if you're looking at a woody stem, right, because that was what we just looked at a moment ago, was one that is more based in herbaceous or non-wood stems like the monocot. This is going to be the eudicot or the dicot stem, right? And so if you look in here, you're going to see that cork cambria, which ends up being here, that becomes the outer bark, remember? And then you also have that inner part, that's the vascular, that's kind of on both sides there. And then you can look, and the pith is here in the middle where we saw it slightly different location before. The xylem is around here. And then you can see basically what they're looking at a little bit farther out is the secondary phloem. Which you can also see right here. And that's part of the overall thing called the inner bark, is towards the inside, and that hard part that you normally can kind of, if it starts separating off, is the outer bark. So this is what it looks like for the dicot. Now, when you have a kind of one of these eudicots or dicots growing, you can actually get what are known, that we see as the small growths off of it, called a twig. And so these are the woody stems. What we were kind of looking at was a big tree that they cut off, but these are more what we're looking at. And so we can compare it to what we saw earlier when we're looking at the herbaceous stems. And if you realize, there's a lot of similarities. Remember how we said we have buds, and these are those lateral buds or the axillary buds that are down, and then you have the terminal bud that's here, the apical bud. It's the same thing. And then between the buds are the internodes, and then the buds themselves, those are the nodes. And sometimes underneath these, you can see leaf scars. And they can tell you how basically the newest growth is on the end that's closest to the terminal bud. The older growth is closer to the root. And these lines that you see here are scars that are left behind by these terminal bud scales or previous winters, where they've cut off the nutrients and basically allowed the leaves to fall, and then they protect themselves there. Now, when you grow off of these buds, there are different types of leaves that you can get. And so leaves can be divided into several different groups. The main groups are listed below, and we have a picture guide. So leaves are important because they're the main part of the plant that carries out photosynthesis. What's really cool about leaves is that their size, shape, color, and texture can be highly variable. And these characteristics, these variable characteristics, are fundamental to the plant identification. Just to remember, the stalk of the leaf is called the petiole, right? The vascular bundles of the leaf make up the midvein. And so when we're looking at here, the surface of the leaf, that's where actually you're going to get photosynthesis, and that's going to take place in the leaf blade. That's the leaf structure that we looked at earlier. So these are the different leaves. So you can get where you just have this nice one single leaf that comes off, and that's called a simple leaf. Compound leaves, as you see, both of these are called. These are leaves made up of many different smaller parts or leaflets joined together. A rachis is another term for the midvein. The palmate veins, these are the veins in a leaf or a leaflet converge to one common point near the petiole of the plant. And so you can see on here that I have what looks like to be kind of. So when we look at here, you can see the palmate compound. You have a lot of them, and they all come together at the single area and they come out. The pinnately compound means that you have them, but they come off this. There's only one of the petiole, sorry. This one, you can see that petiole that's here and that all the leaves are coming off of that. So these are kind of the different kinds that you can look at to get a good idea of how they're working. And this just kind of shows you again to give you a good idea. Petiole here. One comes out of it. Pinnately compound. You have one, and all these are just kind of feathering out. And then this one comes out, and they star all out. Now, just a reminder of the leaf anatomy, of course. You have the apex at the top here. The margin is at the end. The veins are the lines that you can see on the underside. The midrib goes down the middle. And then this is the base. That's where the leaf starts. All of this together is the blade. And then when you add the petiole to it, and then you look at the axillary bud that's here and that little stipule that's on the bottom there, that is that whole structure, and this is what we call a leaf. Now, if we want to actually look at the leaf and we want to cut it in half so we can see what's there, you have on the outside what's known as a cuticle. And the cuticle is important because it's going to be waxy. And it's like this protective layer that they have over there. Now you can see within the mesophyll, well, we have the layers, right? You have the upper epidermis, that's on the top. the lower epidermis on the bottom, and then you have this middle area that is the mesophyll. So in the mesophyll, you'll see you have parenchyma cells. And so the palisade parenchyma cells are actually going to be what have the chloroplast. You can see that they are pretty. Their orientation is pretty rigid, very organized. And then you have the spongy that's below that is very unorganized. They're irregular shaped. They're not as nice and rectangular as the palisade. And they help to facilitate the circulation of air and gas exchange. Now, in that lower epidermis, you'll notice a few things. You'll notice that they have what's called a stoma. And so stomata are cell structures, and you can find these in tree leaves that involve basically in helping to exchange carbon dioxide and water between plants and the atmosphere. And what regulates that are those little guys called guard cells. They're really neat. You can see them, and as they open, they really do look like little mouths kind of opening and closing. And this is important because we need to make sure, especially, and you would have learned this in General Bio 1, the different kind of how they're arranged on the cell differently based on if they're more in, an area where they get plenty of rain versus desert. And so now that we've gone through this kind of overview, what I'm going to do is we are now going to go through and answer some of the pre-lab questions. And then we'll go over the lab one by one. So, what are the three major innovations of angiosperm? And I'd almost say there's four, but I put one of them together. So we have the fact that they're vascular in nature. Is one. That they are flowered is two. And that I said they had fruit, enclosed, seeds, which can almost go with flower too, and endosperm. Now, we didn't talk a lot about their reproduction or their life cycle more so, but do you think angiosperm exhibit alternation of generation? And hopefully you realize that I told you with the dominant plant type and that we're still in plants that it does. And the one that it most commonly or is most dominant is the sporophyte. Now, what characteristics would you find in an angiosperm but not a gymnosperm? So we would find fruit and flowers, right, in an angiosperm. And we would also find things like seeds that are enclosed. And they're enclosed within fruit, right? Within an ovary, that's usually a fruit. What are the two main groups? And they asked this question twice, so I just kind of took one away. And so it is monocot. And if you remember, mono coming from one, and the cot coming from the cotyledon. And just remember, it's a single cotyledon. And that is where the embryonic leaf and the seed-bearing plants will first emerge from in the germinating seed. And then you have the dicot or the eudicot. And remember, the eudicot came from that it is, or dicot, right? EUDICOT, that it's two cotyledons. Name the three kind of plant tissues. It's what we were talking about the whole time, right? We have the dermal tissue, which makes up the protective outer coat. Things like the epidermis or the guard cells, the root hairs, all of this is dermal. You have vascular. So when we were looking at the xylem and the phloem, xylem carrying water, phloem carrying organic compounds like sugar, that's it. And then you have the ground. And the ground tissue is the three different types of cells that we talked about. They're important for the internal organ of the plant. They perform various functions. So you have the parenchyma, which is the cells that carry out photosynthesis and store carbohydrates. The collenchyma, which are cells that usually have thicker cell walls and provide structural support in those regions that are still growing, right? And the sclerenchyma, which is usually dead at maturity, and they make up the woody part of the plant. And basically, what's the point or the purpose, the function of vascular tissue? And this is to move substances. So with that said, let's go in and start looking at what we're going to do for our lab today. So the very first thing we're going to do is we are going to look at the root, and they want you to be able to identify parts of the root using models and so on. So I've kind of gone through here. This wasn't on the initial picture, but I wanted you to get a good idea. So they're going to have you observe the root tip on display, label the root cap, the zone of cell division, the zone of elongation, and maturation, and you can refer to this guide as the model. And so that should help you a little bit. So you're going to take it, the picture that's on your lab, and you're going to basically label it. It's going to ask you what is the function of the root cap, and we talked about that in terms of, protection, and then the cells in the apical meristem divide by mitotic division. What do you think you would find the root apical meristem on this model? So they're asking you to identify it on the model, which I hopefully have helped you with a little bit. And where do you find the root hairs? So I'd say root hairs, remember that is a part of the dermal tissue. So you would look at the epidermis to help with that. Then it's going to get to the next part, and it's going to observe an aerial view of the model. And so they're going to ask you to label the epidermis, the cortex, which is the ground tissue, the parenchyma cells, which will be within the cortex, the vascular cylinder, which is where you will find the xylem, the phloem, and the endodermis, and the pericycle. So I've helped label this a bit. It has some of the stickers because they're hard to see, so I kind of redid it to make it a little bit easier. And so they're going to ask you, what is the function of xylem? And hopefully you realize that it is to help with the flow of H2O. And so on here, they're going to ask you, do you see three tissue types in this cross section? So let's a reminder of what the three tissue types are. They're dermal tissue. So do you see something like the epidermis? Vascular tissue. Do you see something that is composed of, do you see the xylem and do you see the phloem? And then ground tissue. Do you see one of the many types of cells? And I would say, can you see where they have parenchyma? If so, then that question is yes. Explain how the structure of the xylem helps in this function. And so I'm going to have you think about that a little bit. The next thing it's going to do, and this is just some more to kind of help, but then they're going to ask you to go insert the slide of the dicot root. So what you can do here is you go, okay, and sometimes this isn't apparent, and so this is what they want you to do when you're in this. And this is why I also add stuff to the beginning of it so you can see. So you can come back here and go, okay, this is a stem, we don't need this. Let me go back to my picture. Okay, these are the two cross sections of the root. I know that to the right is the monocot and to the left is the dicot. So they want me to draw the slide of the dicot root. So I know that that dicot root is going to have an X in it. So let me go to the part of the lab now, because this is all my added part, right? Everything before the pre-lab is what I've added. And then you're going to go, okay, it's this. I see the X on here, right? And they're going to have you draw it on here, and they want you to label the epidermis, the cortex, and the vascular bundles, right? And so in here, just to let you know, right, epidermis is on the outside, and when you're starting to look at the vascular bundles means they're looking at the area that has the xylem and the phloem. The next thing they're asking, and we're still on roots here, all roots can be classified into one of three types, right? The tap root, the fibrous root, the adventitious root. And so they're asking you here, taproot and fibrous root systems are shown in the root lab guide. Examine and sketch different types on display. Label each diagram with the proper root type. So as you go through there, yes, they have something here, kind of a little creepy to me. And so they go through, and so they have these roots here. I'd probably use this one because I think this one's the best. Or going over here. Each one of them are a little bit different, but depending on what you find the best, right? I like how they have the adventitious one here. So this gives you an idea of, the taproot, that you have the main thick one that goes down and some fringes off of it. Fibrous looks like just a bunch of them all together, kind of like it has a really thick mane of hair. And the adventitious ones are kind of growing out of the stalk that's across the ground, and you can see them at the top, right? And so you can definitely look at these and figure out which ones you have. And you can absolutely look at these and draw them and just make sure that you know the easiest way to see or to be able to identify them later. So the next thing that we're going to look at is the rhizobium, the gram-negative bacteria, and its symbiotic relationship with legumes. And just remember that these both work together to allow each plant and bacteria to get the nutrients they need. The bacteria essentially fixes nitrogen to the roots of the plants. That helps the plant grow, in turn provides the bacteria with sugar and nutrients. And so what you're going to do is you're going to look at these, and you're going to determine basically what is the root nodule, what is the plant root, and what is the bacteria. And you can go and look at here and say, what is the infected zone? Is it this over here, or is it something like this? So you have these two things, and you can kind of look at, do a little bit of research and see what bacteria that have infected a plant root kind of look like, and see if they kind of see what this structure looks like in terms of what bacteria looks like that have infected plant roots. And so it could give you a couple examples. And so then it's going to ask you what type of symbiosis between the bacteria and the plant, what does the plant get from the bacteria? What does the bacteria get from the plant? And how can you tell that the rhizobium is gram-negative? And I can give you a little bit of hint there based on the fact of what you use to stain. And so the next thing that we're going to look at for table 2 are the stem. And a stem is an above ground structure that's going to provide support and facilitate the conduction of water and nutrients. So like I've said before, stems can be herbaceous, which means there's no wood. So if you see a dandelion, it's a perfect example of a herbaceous stem, like in monocots generally, and woody like in the stems of dicots. Herbaceous stems can undergo primary growth, which is the growth in length resulting from the activity of the apical meristem. Remember, at the top, located at the terminal bud, at the very top. There is no increase in the girth of the plants like the stem of the maize or grass. So we're going to look at this model. You're going to observe the model, and this is a monocot stem. You're going to label the epidermis, which think is the outside here. So you might think of something that is in the vicinity of the outside, right? The ground tissue, the parenchymal cells, which would be these guys that you're looking at here. The sclerenchymal cells, which are out here. And then you're looking at the phloem, the xylem, and the vascular bundle. So this whole thing is a vascular bundle, and the small one there is the xylem, and this kind of upside down triangle is the phloem. And you'll notice that the vascular bundles are randomly scattered throughout the stem. So it's a very important feature that is a little bit different than what you would see in a dicot. So which tissue, xylem or phloem, is closer to the epidermis? So you're going to see and locate where the epidermis is, and then look at where xylem and phloem is, and that should give you an idea. What is the function of the sclerenchyma around the vascular bundles? So this is where you go back and you ask yourself, what was the point of that tissue anyway? And they said they normally find it at maturity that make up the thick, woody parts of a plant. So is it there as perhaps a means of structure or something else? And there's one just closer so you can see it. And here is an actual histology of it. So, or we should say histology or cytology of it if we're looking at cells, but it's plant tissue, so histology. It's a zea mays, which is a monocot stem. Examine the sketch and sketch this and label the epidermis again on the outside. It doesn't point, I just put it there. You're going to do the vascular bundle, which is that whole thing, the ground tissue, and that is it. And I kind of show you where the other ones are a little bit easier to see. And they get very close up, so you want to be able to label these. This is a herbaceous dicot stem. So this is an example of something that would be in a sunflower. So they want, again, on here for you to locate the epidermis, which you should know is always on the outside. Hopefully you figured this out. The ground tissue, that is called the cortex, and the pith, the vascular cambium, which is a form of a meristem, right? Vascular bundles, and the xylem, phloem, and sclerenchyma. So some of these are absolutely identified for you, very easy. You can see the circle area right there is the xylem, and the phloem is over here. You can see the ground tissue throughout. The vascular cambium is over here. right? And so they just want you to know. And they're having you practice this and look at this because these are things that you should be able to identify for the lab practical. Now we're going to look at a slide of this. Pretty, isn't it? There's a few of them there. So what they want you to do is to take the model that you just had and apply it to this slide and identify the epidermis and the cortex, the pith, the vascular bundles, the vascular cambium, xylem, phloem, and the scleroderma. They're going to ask you on here, how are the vascular bundles arranged differently in this than they are in the monocots? So look at, are they more structured? Are they more formed? Do they seem to have more order? How are the vascular bundles similar in the monocot and dicot? So when you look at the bundles, are they arranged with the phloem and the xylem in a certain composition? Which tissue transports water? Hopefully you already figured that. And figure out which we talked about a little bit, the function of the scleroderma. We're on table 3, which is the dicots or the woody stems. So in here, they're introducing dicot stems, which are generally wood with secondary growth, which means these can actually, the primary growth is lengthwise. This includes girth in addition to the length or primary growth. So here, the lateral growth is related to the lateral meristem, and that's composed of the vascular cambium and the cork cambium. And secondary growth in the dicots occur because of the activity here, specifically at the vascular cambium. And so this vascular cambium is going to actually produce new xylem, which is secondary xylem, and phloem, which is secondary phloem, every year. And the growth is also called wood. Who knew, right? In addition to the vascular cambium, another lateral meristem is called the cork cambium, and it develops in the outer cortex and replaces the epidermis of the dicot with the periderm. And so what they want you to do is compare the image that you have in your book, in your lab handout, and observe the vascular tissue growth in rings. Label the bark, the annual rings, the sapwood, the heartwood, inner dark, and refer to here. So you're going to look at, in order to do this, because it gives you a big piece, you're going to have to look at several pieces of wood that has been labeled so that you can get an idea of what's going on there. Next, they're going to ask you to take your twig and sketch it, and then label a node, internode, terminal bud, axillary bud on the twig. So you can take this or what I had earlier to show, and you can kind of see. So anything that you have coming out like this, right? Oops, it's hard to see on here. Here, right, or here. Those are your nodes. So the area between those are the internodes, right? And when you look at the axillary bud, you're looking at something off to the side, right? Not something on the top. And so they're giving you nice. And now we're going to go to leaves. This is important for the whole process of photosynthesis. So it wants you, there's several leaves, so these are leaves that they have, and they want you to identify the leaf blade, the petiole, and the mid-vein, and the rachis, and the compound leaf, the mid-vein, and the simple leaf. So simply draw the leaf, a simple leaf. So you look at here and say which one of these are simple, and then you're going to label what they ask for. Then they're going to ask you to go to the second set. What kind of leaf is this? Is this pinnately compound or palmately compound? And so you need to identify the difference between those two species. You're going to draw them, and then you're going to label them. And they want you to sketch and label the veins. And then they have pictures for you to go through to help a little bit. And then looking at the veins between each up close. It's very nice pictures. And the last part is leaf anatomy. So they're going to start with this is a dicot, eudicot leaf model on display. And so they want you to take it and they want you to label. And so I've also provided pictures earlier. So they want the upper epidermis, which is going to be towards the top, away from any of the stomatas or guard cells if you have them. And you can identify if you have the upper or lower epidermis by the parenchyma cells. So look at where the pallosides and the spongy are, and then you can do a really good reference to that. And remember, the pallosides ones are the ones that are really ordered, and spongy, like if you have spongy bone, is just kind of all over the place. And then we're going to also want you to label the vascular bundles and the xylem and the phloem in here. And you can use here any of these to draw. And they have a picture on the one that they want you to use originally, which is this one right here. And then they want you to answer, describe the orientation of xylem in the vascular bundle. Is it facing the upper epidermis or lower? What is the function of the cuticle? What's the function of the mesophyll? So think when you're thinking about the mesophyll, what's located in the mesophyll? Are there specific cells there? I can think of a couple cells that we're looking at right now that are located in that section, and they're really important for energy. Does the leaf have vascular tissue? If so, where? And you can go and kind of take a look at the first couple slides I had. And where would you find more stomata on a dicot leaf? In the upper epidermis or the lower epidermis, and why? Then it's going to take you to the monocot, and it wants you to label the epidermis, the stomata, the veins, which are the xylem and phloem, and the mesophyll. And then again on here, they give you a really nice way to see the stomata with the guard cells here. And they're going to ask you, what is the difference between the mesophyll? So if you look here, I would ask, do you see really nice organized rectangular parenchyma cells in there? That's what I would start wondering. Do you see any similarities in the vascular bundles of monocot stems and monocot leaves? And what is the main function of a leaf? Give examples of leaves that perform other functions. And then they're having you look at the anatomy here of the leaf. And so how is the monocot leaf different from the dicot leaf? So you're going to go through here and give some examples of how they are different. And then you'll have your post-lab questions. So if you have any questions, please email me. I hope this is helpful. Good luck, and I will talk to you soon.